Hello, this is Lydia Fleischman from NCBI, and on behalf of the team, I wanted to greet everyone here in the Spheres meeting and thank you, Duncan, for having us. We'll be having a couple of people from our team speak today about sequence submission, specifically of SARS-CoV-2 sequences, both unassembled and assembled, as well as a great annotation tool, VADR, which Eric Naraki will be speaking about. And then we also just wanted to kind of emphasize the dialogue that we want to keep open with this community and with the greater community. Eric, if you could go to the next slide. We want to engage with you and really continue a dialogue on NCBI submission. We want your feedback on our existing processes and data and tools. And so we really welcome this opportunity to do a refresher of what we have for SARS-CoV-2 submission to SRA and GenBank, introduce that helpful tool for annotation that I mentioned, and share a little bit about ongoing opportunities for feedback and interaction with the NCBI team. And with that, I will pass it over to Eric.
Okay, thanks, Lydia. So my name is Eric Naraki. I also work at NCBI. I'm gonna be talking about VADR, which is the software tool that I'm the lead developer of that is used for SARS-CoV-2 analysis. So we use it to validate and annotate incoming sequences. So it's a general tool that you can really use for any type of viral sequence. And it does the annotation and validation based on a reference. I'm going to go into detail as to how it works, but at a high level, it does this by aligning sequences globally with respect to the RefSeq. It can also use a profile model. For SARS-CoV-2, we just use a single RefSeq, but it's designed to use profiles also. So those are models built from an alignment of sequences, representative sequences, instead of a single sequence. We originally tested it for dengue virus and norovirus, but it's been in use for SARS-CoV-2 since March on a kind of manual base. It was being used manually early and then switched to a more automated pipeline in June. So this work was published in BMC Bioinformatics earlier this year. Okay, so to kind of introduce the motivation for why we developed VADR, I'm going to talk about kind of how sequence submissions go just very briefly. So at NCBI, at GenBank, we have indexers. Linda Yankee is one, Vince Calhoun is another, who deal with SARS-CoV-2 sequences. And they take the incoming sequences and metadata from the submitter and are in charge of determining if they should be accepted into GenBank or if there's if they should be returned to the submitter for further validation or because there's some issue with them. And so we developed VADR as an annotation tool for viruses, specifically to help the indexers. So we know that there's other viral annotation software out there, some excellent software that we could have used. So Vigor as an example, Viper, Vapid. These are really great tools, but we decided to develop our own software because we wanted very precise control over which sequences would automatically be let into GenBank versus, and we call those passing sequences, versus sequences that have to be looked at by a human indexer. So it's really important that we don't want to let any sequences that are bad automatically into GenBank. So VADR identifies many different types of issues with sequences or kind of unusual features of sequences, some of which may be innocuous and the sequence is perfectly biologically valid, but those situations warrant the sequence to be looked at by an expert indexer. So that's what VADR does. It passes some sequences automatically and others are gonna fail and be looked at by the indexer. And the indexer then looks at the information returned from VADR and determines if those sequences should be accepted because they look fine or if they should be returned to the submitter. And I'm not showing another arrow here, but there could be an arrow here where VADR determines that some sequences are kind of so obviously messed up in some way, that they should automatically be returned to the submitter without an indexer having to look at them. So some kind of obvious problems. So now I'm going to go into kind of how VADR works. And the first step of VADR is to build a homology model of the virus you're trying to annotate and validate for. So we do this using RefSeq currently. So this is an example using dengue, but everything I'm going to talk about applies also to SARS-CoV-2. So these are all of the features in the GenBank record for one of the RefSeqs for dengue, the type 1. And I'm showing you a CDS feature, the gene, which is essentially the same as the CDS as far as coordinates, the MAT peptides that the CDS is broken down into, the mature peptides rather. And there's also secondary structural elements in the dengue genome that are that are features in the RefSeq, and those are actually modeled by VADR. So when we build a model for VADR, we build a profile model called a covariance model that includes both sequence information and secondary structure information. And so when it does analysis, it can take that secondary structure into account. These these structural elements, stem loops, come from the RFAM database, and there are a few on SARS-CoV-2 as well. So VADR, I'm going to talk about pretty specifically what VADR does. So I'm going to go through the 4 stages of analysis that VADR does. And on this slide, I'm just going to list them, and then I'm going to go into detail for each of them in the next slides. So VADR annotates each sequence using its best matching model. So it's actually set up to use a library of different RefSeqs and to select the closest one and then use that RefSeq to do the annotation for each incoming sequence. For SARS-CoV-2, like I said, we only have one RefSeq. So this classification is essentially to make sure that the sequence is in fact SARS-CoV-2. Once it determines the best matching model, it then moves on to a stage we call coverage determination to find kind of the local hits against the model and how they align. And then there's a global alignment stage where it computes a global alignment of the sequence of the model and then uses that to map the features, just using the coordinates of the features from the RefSeq, just mapping them based on the alignment. And then finally, we do a protein validation stage where we translate each CDS and then compare it to the RefSeq proteins using BLASTX. And the reason we have these 4 different stages, and I'll go into this more in the next slides, but is that each one of them identifies different types of issues, or what we call alerts in the sequence, and these are going to alert the indexer as to these unusual features of the sequence that they should look at. So in the first stage, we have a library of models potentially where we compare the sequence against each model in the library. In the case of SARS-CoV-2, again, we only have one, but it is set up. The reason I mention this here is because it is set up to use potentially multiple references. So if we did add new references based on common mutations in SARS-CoV-2, you could potentially differentiate between those with VADR. So we select the best matching one, and then that model is used in all the subsequent steps. And so at this stage, I'm now showing you a table that has a list of the types of alerts or issues that we could find with the sequence at this stage. So one of them is no annotation. So if it doesn't match any of the models, that's an issue, that's a problem. Could be that it's reverse complemented because the match is on the opposite strand. And with VADR, you can actually specify what you think the sequence is, and if it matches to a different model, you can specify the type of model you think it is, like dengue, and if it matches to a norovirus, for example, that would be a different type of problem. So in the second stage, now that we have the best matching model, we use a BLAST-N-like algorithm to do local alignment between the model and the sequence. So we actually don't use BLAST-N, we use Hammer 3. But it's the same type of idea. So if we have I'm going to go through some example scenarios. So imagine this S1 sequence here. Let's say that's a full length sequence to the model. We'd expect one hit that is essentially the full length of the model, and that's expected. So there's no alerts reported there. We could also have a shorter sequence that's not full length with respect to the model, but the entire sequence is covered by the match to the model. And so that's just a partial sequence. Again, there's no problem there. But we could have other situations, like for this S3 example, where the match extends, does not extend to the full length of the sequence. So there's extra sequence on either end that doesn't match the model because it's not similar enough. And so those are specific types of alerts that we then report. And so in this case, there's a low similarity on the 5' end, low similarity on the 3' end, and also we'd have a low coverage alert because not enough of the sequence matches the model. So in another case, we might have multiple hits where there's a region in the middle that doesn't match the model. Again, that's a different type of alert. So at this stage, like I mentioned, low coverage, we could have other problems that we identify. We could have the case where two parts of the sequence match to the same part of the model. So those would be duplicate region, that's a duplicate regions alert. We could have a case where the order of the hits with respect to the model is different than the order with respect to the sequence. So you have kind of the sequence in the wrong order. That's a different type of alert. We could have hits on both strands. I mentioned these low similarity issues with the previous examples. So then on the next stage, we do another comparison with the model. And this time we do a global alignment to the model, where we just take the sequence, so going back to the S1 example, and we align it to the full model. And based on that alignment, we can just map all of these features and get the endpoints of all the features very simply. And again, S1 and S2, we would have no problems with. They're perfectly good sequences that we don't see anything wrong with. Now, S3, I showed you on the previous example, we had extra sequence that didn't match. When we do a global alignment, we're actually forcing it to align at those ends. So I bring this up to point out that that's why we also do that other blast N hammer 3 stage, because if we just forced a global alignment, it would be more difficult to tell that there was anything wrong with these ends. So we do both a full alignment and the coverage determination step to kind of identify different types of problems. Again, with S4, this region would be aligned. So it would be harder to tell there was something wrong there without post-processing the alignment. But we could have other situations that we do find problems with now that we're doing a full alignment. So this S5 example, we could find that when we try to map this map peptide boundary here, we find that it's a gap in the alignment. And so we basically are saying we're not sure where that position should be for the end of that map peptide. And so that's a different type of alert. We could also have a case where we have low confidence in those endpoints. So a covariance model is a probabilistic model, and we're able to compute the posterior probability that any nucleotide is aligned to any position in the model. And if that acts as a confidence estimate into how well resolved the alignment is there, kind of how certain the model is that the alignment is correct. And if that probability is low enough, it's basically telling us that it's uncertain where that boundary should go. So that's a different type of alert. We also translate coding regions at this stage and look for in-frame early stops. So there could be an in-frame early stop. That's a different type of problem that we'll detect. So at the alignment stage, there's a lot more alerts we can detect. Most of them have to do with coding potential. So mutation and a start codon, mutation and a stop codon, early stops. We could also have mature peptides that aren't adjacent to each other. And I mentioned these indefinite annotation. That's when there's a gap or a low confidence at the end of a feature. So in the final stage, we do protein validation. And like I mentioned before, we just translate each CDS and then blast it using BlastX against the RefSeq proteins. And then we expect that BLAST X alignment to be full length with respect to the CDS, predicted CDS. And so that in this case, if we had an early stop or a frame shift mutation, we would get a shorter alignment, and that's a specific type of alert that we report at that stage. Oh, and I wanted to mention Alejandro Schafler is a colleague of mine that implemented this stage. So at this point, in the protein validation stage, we identify these early stops. We also report an error if the BLAST alignment does not extend to the ends. And also we look for very large insertions or deletions at this stage, and that's a different type of alert that's reported. So those are the 4 stages of VADR, and that's kind of how VADR works in general. And those are the alerts that we developed when we were testing for norovirus and dengue virus. When we started analyzing SARS-CoV-2, we realized that there were 2 issues that we had to address specifically for SARS-CoV-2. So the first one is the processing time. So the covariance model alignment, I mentioned that it incorporates secondary structure into the alignment. That comes at a computational cost. That's a more expensive algorithm than just sequence-only alignment. And for SARS-CoV-2, it's so long, and that algorithm scales with the more than the cube of the length of the sequence. So it's especially slow for very long sequences. For SARS-CoV-2, it was taking about 5 minutes for a full-length sequence. So that was way too slow for high throughput. So we implemented a heuristic based on a BLAST-N kind of first pass alignment that reduces that to about 2 seconds per typical sequence. So now it's not blazingly fast by any means, but it's still, it's fast enough for kind of the throughput that we're using it for GenBank submissions. So the second issue is that SARS-CoV-2 submissions SARS-CoV-2 sequences are being submitted tended to have a lot of ambiguous ends in them, or in stretches of ends in them. And if you look at this table down here, it's showing you the percentage of nucleotides that are ends in SARS-CoV-2, norovirus, and dengue virus. And more importantly, the percentage of sequences that have at least one stretch that's fairly long. So in this case, greater than 50 nucleotides. And you can see about a quarter of the sequences have a stretch of ends greater than 50 nucleotides. The reason that's so important is because the VADR and other annotation tools rely on sequence similarity. And so whenever there's a stretch of ends, it essentially can't get purchase on that part of the alignment. It has to rely on the bordering sequences. And so it just makes it more difficult to do the alignment correctly. So what we implemented was a method to identify those stretches of ends, identify where they map to in the RefSeq, again, based on a first pass BLAST-N alignment, and then replace those ends with the expected nucleotides, just taking them directly from the RefSeq. Then we do the full analysis using those sequences with those ends replaced, and that seems to work pretty well. So what I'm showing you now is the observed counts of the different types of problems that VADR is identifying in SARS-CoV-2 sequences. So I just want to make 2 points. I'm not going to go through all of them, but the 2 points I want to make are there's many different types of problems. So it's finding a lot of different issues, and some of them are much more common than others. So that duplicate regions is a pretty egregious problem, but it doesn't happen very often. The more common ones are like CDS with a stop codon, early stop codon, for example, or other problems with the BLAST X alignment not extending far enough. So now I'm just going to go through just 2 examples of real SARS-CoV-2 sequences and alerts that VADR reported on them. So this first one was actually fairly early. This was, I believe, a sequence from February. And this had this error, duplicate regions, where it identified that there was one part of the sequence that actually mapped to the same part of the model. Or sorry, 2 parts of the sequence that mapped to the same part of the model. And it's This is the alert. This is the message that VADR reports. It tells you the coordinates of where those, so this is the region that overlaps. And it tells you that information so you can then go and look at it. It also identified that at the beginning there was a stretch of 24 nucleotides that didn't match to the model. And again, at the end, there was a stretch that didn't match to the model. So now I'm going to show you an alignment of those regions. Yes.
This is Ryan. Lee Katz in the chat asked a question that I think you could clarify quickly, which is just when you're substituting the RefSeq sequence for the ends and the submitted sequence, that's just for the purpose of the QA step. That's not making it into the GenBank record, assuming it gets accepted, correct?
Correct. Yeah, I should have been more clear about that. Yeah, so we replace the ends with the RefSeq nucleotides, do the full analysis, and then put them back basically before it goes into GenBank. Okay, so
I apologize, Eric. Can I follow up on that?
No, go ahead.
This is Lee.
That's fine.
I just ran VADR for the first time this morning, so this is fresh on my mind, and it's great.
Okay.
And so when I ran it, it produced that table file, which means I could take my genome assembly and upload that to GenBank with those annotations. Is that possible that those artificial annotations could make their way to GenBank just by what my workflow was?
So I guess you're talking about the boundaries of the annotation, like, so do you mean like if you had a stretch of ends at the end of a CDS that the boundary would be be an N? Is that what you mean?
So like the boundary of the gene might be artificially kind of guessed on that QA/QC step, but that might also make it into the table file, which I would then upload to GenBank.
So there's rules in GenBank that, for example, CDS cannot end with an N. So those positions get what we call trimmed. So they get modified to the 3' most, like if it was at the end of the CDS, the 3' most non-N. And then we add this like greater than sign that indicates that, it's not to the end of so it's partial basically. So I don't know if that answers your question. So VADR should generate the annotations like with that greater than sign instead of saying that it's a full length CDS. Does that answer your question?
Yes, I think I'm understanding that I might have some, it's possible I have some noise in the table file locally, but the GenBank submission process would catch that.
I believe that's right. So there's additional steps that are done to go from the VADR output to the final files that go into GenBank. Linda Yankee can speak better to it than I can. So maybe we could bring that up with her, and also you can email us and, give us a specific example and we can look at it also.
Thanks, I appreciate it.
Sure, no problem. Okay, so for this first example, I'm now just showing you an alignment of like what that looks like in the actual sequence data. So at the 5' end, there was a problem where this sequence was not similar enough. And that actually matches to the end of the RefSeq. So there was some problem with perhaps the assembly of that sequence. And again, at the end, so this is now the 3' end, this region matched best to the middle of the RefSeq. And that was actually reported by VADR, that it identified that region was duplicated. So that's one example. In that case, the sequence was trimmed by Linda and it then went in. So those ends were trimmed because they were deemed to be not accurate. So for the second example, this is a more recent sequence. And in this case, yes.
So going back to the previous one, the low similarity end. This is Pavitra from University of Washington. Does it also call an error for low similarity end if there's a differing number of A's in that poly A at the end? How sensitive is that?
Yeah,
a lot of ours get called out for that reason.
Right. So that's something we've talked about. So the end, it will, yes, it doesn't treat that end any differently than any other region of the alignment, although it could. So if there is like, if it doesn't match well in this poly A track, it could potentially report an error. And that if that's something that's happening a lot and it's biologically valid, that's something we can look at to try to like come up with a way of dealing with that specially. So if you have a lot of examples of those and you want us to look at, please send them.
I just feel like we shouldn't penalize just for having like 3 A's at the end versus like, the exact number as the RefSeq, because I guess, like
Sure.
Your assembly might just generate those many, and we try not to introduce like additional things from the reference for obvious reasons, but like that's just what the assembly came up with.
Right. Yeah, no, I think I understand. It's just like, because it's treating that the same as any other part, it's going to throw an alert. But like I said, I agree with you, and it probably shouldn't. So if you have examples of that and you could help us out by sending them, we'd be happy to look at that.
Sure, will do.
Thanks.
Thank you.
Okay, so for the second example, it was just an early in-frame stop. So this is in ORF 8, I believe, which is commonly has issues like this, where there was a stop, 342 nucleotides, 5' of where it was expected to be based on alignment to the RefSeq. So it was an early stop, and this was detected twice, both in the alignment stage and then again in the protein validation stage, but they're both saying the same thing, although it's 2 separate alerts. And then if we look at the sequence, this is an alignment of ORF 8. Just a part of the alignment. It starts at these positions. This sequence is starts in the second nucleotide of the RefSeq, so these numbers are off by 1. But it's completely identical except for this one mutation here, and it's changed a GGA in the RefSeq to a TGA, or not. Yeah, so it's introducing the stop, and that's what it's detecting. In this case, Linda went back to the submitter, who kind of verified that it was correct, and this sequence went in as it was. So this is an example of a VADR problem that turned out to go into GenBank because it wasn't actually a problem. Okay, so I'm going to wrap up in the next couple slides. So I just wanted to mention some limitations of VADR. So one of them is it works in nucleotide space. We did that mainly so it could do global alignment of the full genomes. But as many of you know, if you work in protein space, you have more statistical power because of the larger alphabet. And, we're not working in protein space except for the BLASTX step. So that's a limitation. You are really, your RefSeq or your model really has to be representative of the sequences you're trying to annotate. If you have divergent sequences that have novel stops, like that early example, they're gonna fail. And so you really want to make sure you have kind of coverage in your model of everything you're expecting to allow as valid. This is a practical one. So I mentioned the slow runtime of covariance model alignment. It's also very memory intensive. So that heuristic that we implemented to get the speed down to 2 seconds also reduces memory time, but in some cases, memory usage, but in some cases, that in rare cases, like 1 in 1,000 sequences empirically, that doesn't really work. And so you have to fall back to the expensive CM alignment. And so you do need 64 gigabytes of RAM to run VADR for SARS-CoV-2, and that's a big limitation. We realize that. Wanted to point out some important features. So like I mentioned before, this pass-fail decision is really important to us, and the indexers or users have precise control over that. So you can change which of those alerts are fatal versus non-fatal, so cause a sequence to fail or not, as well as the thresholds for many of them. we do have so VADR's VADR's on GitHub. There's a standalone version you can download and use for submitters and also for other purposes. So if you're just interested in doing analysis of SARS-CoV-2 without submitting the sequences, there is a separate, we think, potential pool of users that would use it for that case, and also other viruses. You can build your own models and use it for them. It's a general tool, but again, you really need the models to cover all the diversity that you're expecting. And the way that the alignment feature mapping works is really flexible. So you can have a feature as short as a single nucleotide and potentially multiple segments. And that's what allows us to handle things like program frame shifts where you have a duplicated nucleotide in a CDS very easily. So just want to mention there is documentation on GitHub. I made a special page just for SARS-CoV-2 analysis. It's kind of a tutorial of how to do this, or how to use VADR for SARS-CoV-2. And then there's, in the source code, there's markdown files so vannotate is the script that's used to do the annotation, and it has all the command line options and everything documented. Okay, and so I think I'm gonna pause now and see if there's any other questions. Since I can't look at the chat, are there any other questions?
Hey, Eric, this is Eneida. Just none other in the chat unless anybody else has any that they'd like to volunteer.
Okay, thanks, Eneida.
Hey, this is Jake Garfin at the Minnesota Department of Health. I was just wondering if the parameters that are used on NCBI to pass-fail sequences are available either on GitHub or somewhere else?
Yeah, so in that documentation on this page, I wish I could click on this link, but yeah, I think this second one, it does show all the parameters, and that's a good question because there is specific parameters that GenBank uses, and that is available. Yes.
Okay, great, thanks.
Yep.
Hi, this is Danny. Sorry, I missed the part at the beginning where you probably talked about what the output was. Was it like sequin or ASN format?
I did not talk about that actually, so you didn't miss it. So the output is a sequin table file. It's not ASN.
Oh, so it's something you feed into TBL to ASN?
Correct. If you're doing it by yourself, yes, that's an additional step that needs to be done. If you're submitting it through GenBank, they automatically do that.
Hi, this is Pavitra again from the University of Washington. Again, I also missed the first part, so apologies if this was covered. But if for those who are submitting via the web, the web submit.ncbi, is it at all possible to have some sort of machine readable output format for that rather than have an HTML report so that we can like subset sequences and stuff,
Right. So we're open to requests like that. So so that's something for not just me to answer, but yeah, if you have a request like that, please send that in, and maybe Linda can speak to it.
Actually, I have a slide about that later. That will be in the next version.
Yay! Thank you.
At least an attempt. We'll see if you like it or not, and we can go from there. But our first stab at it should be in our next release.
Okay, thank you.
Okay.
Hey, this is Lee Katz. I wanted to follow up on Danny's question earlier. It produces a table file, and I don't know if this is outside the scope of this talk, so please let me know if it's outside. Is there any way to get GFF output for things that use GFF for pangenomics, or if I just wanted to do some kind of rudimentary allele calling or something, it might be easier to use GFF?
Yeah, that doesn't exist now, but it can definitely. So I will work on that so it outputs GFF.
Thanks.
Yeah, for now, there's a output file .ftr. It's like feature information. If you need it earlier than that, that's probably the easiest file to parse to get to GFF.
Thanks.
Yep. Okay, if there's no other questions, I will hand it off to Linda Yankee, who's going to speak next.
So just a quick recap of what VADR and how we use it for submissions. First and foremost is speed. Every submitter will tell you that the hardest part of submitting data to GenBank for them is adding the annotations. And by providing this tool that not only you can check your data before you submit, but also we'll run the tool for you and add the annotation for you. Saves you a lot of time, and it also results in data being released to the database much faster. The quality control aspect, VADR checks the sequence in both the coding and non-coding regions. And if you've looked at a lot of DNA analysis, you know sometimes catching anomalies in non-coding regions is harder to do. The consistency, providing the standard nomenclature for both the genes and the products, that's actually an extremely important asset for data retrieval and doing comparison studies. And also there's value added. Most submissions that come to us, even of viruses, don't contain all of the mature peptides or the RNA structural components that VADR actually will add for us. And note that it does do the mature peptides on both of the polyproteins in SARS, which can be a little tricky since they're duplicated. Next slide, please, Eric. Eric?
Sorry about that.
Thank you. No problem. So we do have two mechanisms to submit, and we've been talking a lot. I notice I'm going to change this up a little bit because of the questions that were asked previously. And both of the submission protocols that we have right now, the common requirements are FASTA, the source metadata in a table format, and submitter information. You do not need to run table to ASN yourself. You do not need to supply that feature table that's output for VADR. Again, VADR is great if you want to check before you send to us, but you do not need to supply that to us. We will run table to ASN and run VADR for you. We have both a web-based interface and a programmatic interface. I believe the last time we spoke, we talked about the web-based, but the programmatic wasn't available yet. They're both up and running. And in both cases, you can supply the bioproject, the biosample, and the SRA run accession, or the SRR, so to speak, in the source table, and it will be picked up and added to your records to provide that link between all of your data. While the way that you submit the data is different, the reporting system is the same. So for both of them, we gather the files that are supplied, run VADR to get the feature table, run table to ASN, and then output either the error reports or any kind of exception reports, all of that to the web for you. Next slide, please. So I'm going to talk a little bit about the web-based submission first, because that's what we seem to be getting the most of right now. Basically, it's forms online that prompt you for the required information. The source information that you supply can be a table that you already have and you import, or actually within the program there is an editable table so that you can copy, paste, move things around, and so forth as you go. A big advantage, in my opinion, of using the web-based form is, particularly for smaller groups of submission, is that there's real-time validation of certain things like the country date isolate format or sequence length and vector screening. You can see this is actually a snippet of one from influenza. That's why the error is about serotypes. But the general idea is you'll get a warning or an error if something is wrong right away while you're actually in the program, so that you can make any changes you need to make at the time. Errors need to be fixed before you can move on. Warnings are for things like, hey, you only put the collection date year, and it would be really nice to have more than that. So that way we can try and get as much information as we can. Next slide, please, Eric. So the programmatic submissions are, if any of you, and I know there are people on this call who have used the same system to submit influenza sequences in the past. Essentially, you upload a zip archive and a submission.xml file via FTP. The archive contains that day, the source. The SPT is the submitter information. And you can also supply an optional comment field, file that will show up in the comment field in the final flat file in the programmatic submissions. You do need to include that submission.xml file. It's basically the directions that tell us where this data is supposed to go and what pipeline to use. Again, there's some basic sequence checks that are done as soon as we receive the files, the low quality vector, sequence length, the format. If all of that and all the files are there, essentially you get a temporary sub number the same way that you do if you use the web-based form. And that will be assigned and available in the report.xml. Then we'll go through again, run VADR, run table.asn, and any accessions or files will be posted on the submission portal for these submissions as well. Next slide, please, Eric. So what I have here is what is 2 successful submissions. This was in our test system, so all of these are made up. The first one is using the web-based pathway, and the second one is using the programmatic interface. You'll notice that the output is basically the same. You get the accession numbers. You get a tab delimited file that you can download that has the relationship between the accession number and the seq ID or sequence identifier used in your original submission. You can also download copies of flat file and the email that we send to you when it is released. If you did not put a hold date on your sequences, which I hope most of you are not, this will be a live link that will take you to the records in the database as soon as they appear. Next slide. If there is a VADR alert, you'll get an error report. Again, the top one is a web-based form and the lower one is a programmatic. And you can see that there's a link to an error report. Again, it says error, not alert. It could be just an error for us just means that someone needs to look at it. So if you were to click on or download that report and open it, you'll see this, which is a list of the errors as they appear and the sequence identifier for each error. If you click on that link, it's basically just an explanation of the error. In some cases, we can give you hints as to how to correct it, not necessarily for a stop codon, but for other errors we can. If you'll notice, the submissions that came in with the web-based tool have a fix button. If you click on that button, it will open up your previous submission at the point where the error was detected. So for example, if there's a problem with a stop codon, it's going to take you back to the page where you uploaded sequences and give you a chance to upload an amended file of sequences that corrects that error. Because the programmatic interface doesn't have a web submission, we can't offer that. In order to fix those submissions, you will have to re-upload the archive in the submission.xml file. Next slide, Eric. So, future improvements, what we were discussing. Just as Eric made changes in VADR to handle the ends, we've been making changes in the validator as well, and this will result in less sequences being flagged for manual review and more will immediately flow through to be released to the database. We also want to include another error report, so we'll still have that basic simple one, but we'll have the more detailed alt file that is produced by VADR. A little snippet of the one that is generated for the example. This is the same sequence that I showed the current error report for, but you can see it says CDF has stop codon just like before, but now you at least find out which protein has the stop codon. And it has a lot more details in cases where there's a difference in the alignment. The actual nucleotide positions are shown in this, and this will be something that you can download and parse out and use to QC the files yourself. Okay, next slide, please. So this is, as we were saying earlier, VADR is going to alert on detected frame shifts or stop codons. We know viruses mutate. We know that a lot of these are actually correct. We've gotten a number of confirmed mutations so far in the genes that are listed at the bottom of the slide there. We can, as Eric kept saying, we can add VADR reference sequences. So if you know of sequences that have a mutation at a place that is consistent, so let's say ORF7A is always truncated at position amino acid 50, please let us know, and we can find a sequence to add to the reference set. So what this will do is will mean that in the future, if you submit another sequence that has that mutation, it will not throw up a VADR alert, and it will go right through the process without anybody having to review it. So we'd really like this community's help in analyzing these, if at all possible. If you do have one of these sequences where you have confirmed that what is flagged as an alert is correct, please let us know. You can write to GB admin, and you can just let us know the read coverage over the area of the mutation. And if you've not already provided the SRR number, it would be great to have that, because one of the themes that we've had in several of these talks is trying to link the assembled data back to the read data and assess whether there's any anomalies. So we really are trying to promote getting that SRR information. And that will actually lead into Yuriy talking.
Okay, great. So as I was saying, what we want to do is create connections between GenBank and SRA to make it a lot easier for users to either get the GenBank data sets or to get the SRA data sets. It's very common for me to hear a request from users to go back and forth, either to get the raw data so that they can confirm findings that they saw in the GenBank record, or be able to first look at a GenBank record before having to download a large SRA data set and have to spend lots of time assembling it and doing other things, and they would instead prefer to look at GenBank submissions. So if you are submitting to NCBI and you are submitting COVID data sets, we did create specifically an umbrella bio project for all sphere submissions, which you can see on the slide. And if you are starting out a new project yourself, you can enter this umbrella bio project as part of your initial submission to register your own bio project. This way, all of the bio projects submitted under spheres would be grouped together, making it that much more easier to retrieve the entire data set if somebody wanted to process things in bulk. So you can make sure to use the same bioprojects and biosamples in both your SRA and GenBank submissions to make it easier for us to interconnect the data sets on our back ends. Next slide, please. So if you're getting started with SRA submissions and you've never submitted to us before, we are the raw data repository. And as you're submitting to SRA, if you've never submitted a data project, you can submit your bio project and biosamples, which is basically your study and sample registration, as part of the SRA submission. So you don't need to do this piecemeal if you have all of your data together at the start of the submission. We do have a very flexible upload options. We have web uploads through basic drag and drop. We have an FTP upload method, and we can get data from AWS buckets directly. We are working on getting data from GCP buckets, but that's going to come probably next month, as we still have some development work to finish up for that. But there are multiple ways to upload to us. And of course, we have the same programmatic submission pathway as GenBank. So we use the same kind of backend system to do all of the submissions programmatically. So should you want to submit 10,000 or more samples at the same time or over the next year or two, that is definitely a great way to go about it. So you can drive your submissions off of your own LIMS or database that you use for storing and organizing your own internal data sets. Next slide, please. As you create an SRA submission, once it's submitted and SRA has had a chance to process it, we do provide a way to modify the contents of the submission through a web interface. If you go to our submission system, it's called Manage Data, and you have several things you can do. You can create reviewer links if you're doing submissions that are part of a publication, so you can send this link to reviewers so they can take a look at the samples and the descriptions of the data set you've submitted to us and how it applies to your publication. And you can edit your title, descriptions of your bio projects. You can add grants and publication links after the fact of submitting the publication. And of course, you can modify SRA data as well. We don't allow modification of the actual files you submitted. That requires a new submission. But any type of metadata that's text-based, you can modify it through the web portal, and it usually gets processed and incorporated it very quickly, usually within minutes. It might take a little bit longer for it to appear on our web pages, but that just is a matter of getting indexed. And I guess I wanted to throw up some statistics. So we've had roughly 11,000 runs from USA, and overall we have around 100,000 runs from around the world, and the 100,000 does include USA. So we do have a large amount of COVID data that's available for you. And the best part about it is all of this data is part of the AWS Open Data Program. So it's available to download for free. You don't have to be in the cloud to get to use it. You can download it to your local compute farm and process the data there. And we do provide multiple different formats there. So feel free to check it out and let us know any feedback. Next slide, please. So the last slide is we're here for you. We've done lots of things with your input that have made very good improvements to our system, such as the programmatic submission options, the file upload, drag and drop for the system. We've had multiple documentation edits thanks to the feedback we've received from the Spheres group. But we do still need feedback from you guys to continue improving our services. We certainly have some biosample linkages and data flow discussions for GenBank submissions. We've briefly talked about how to do programmatic submissions for SRA and GenBank at the same time. And for SRA, we do have what for now we're dubbing the human scrubber tool for being able to identify human reads in raw data files and being able to remove them, so that if you're submitting data to SRA and you're worried about human data being present there, this tool could help you remove those reads. But we definitely need feedback on that tool. So if you're interested for any of these things, please just contact us and tell us you want to be a tester or provide feedback, and we'll definitely schedule an interview with you so that we can hear all of your feedback. So thank you.
I don't know whether we'll have time for a Q&A. I realize we're at time exactly now, but the good news is I think we will continue to have NCBI updates and discussions. For one thing, I think we can continue some of the Q&A online, but I think NCBI is committed to doing a series of updates over the next couple months on different topics. So I think it'll be a great opportunity to pull back the curtain on some of these and really provide some user feedback, which I think is absolutely critical on the NCBI side. I'd really like to thank Lydia, Eric, Linda, Yuriy, Eneida, Eileen. Am I missing anybody? Great work from the NCBI team. You know, I think this has been very insightful for me. I think just given the quality and number of questions, I think there's a lot of interest in how to work better with the NCBI resources and to ensure that our sequences are submitted and annotated properly. So we're excited by that. Thank you all, and we will talk soon. Lydia or others, do you have any sign-offs or anything you need to add?
Thank you from NCBI.
All right, thank you all, and we'll see you all next week.